This video is meant for GCSE, but it could be used as a precursor for A-level. It's part of the new specification, and so we haven't covered any of this before. Let's look at the objectives. What a nucleotide is, more about the structure of DNA and how it relates to its function, and how DNA controls protein synthesis. So, what do we know already? Well, we know that DNA is the code for making proteins. We know that DNA isn't just for inheritance and making gametes, that we take the genetic code for each protein from it. We know that ribosomes are the site of protein synthesis and that ribosomes exist in the cytoplasm. We know that amino acids are a polymer of repeating units that make a protein. We know that the manner in which the protein folds is important for its function. And we know that a different sequence of amino acids would make it fold in a different way. And if it folds in a different way, it may have a different structure and therefore it won't fit on the receptor or it won't have the right active site. So this is a new idea really, because when we've thought about DNA before, we've always just thought about it as something that makes up chromosomes that is important for making gametes and for inheritance. We're now thinking of that DNA as a code for making proteins. So let's look at the molecular structure of DNA. This is a worksheet I've used with my classes. DNA is a double-stranded helix. This shows the helix unwound and it shows one of those strands. The first thing the worksheet asks you to do is to circle a nucleotide. So a nucleotide is a phosphate, a sugar, and a base, and it could be any of that one, that one, that one, that one, that one, or that one. The nucleotide is the monomer that makes up the polymer DNA. The second thing asks you to colour all the bases in blue, so that's those ones there, and look at the shape, how the shape matches together. So you can see that C will only join to G and the A will only join to T because that's how those shapes fit together. The next thing we're asked to do is to colour in the sugar red. And here's the sugars, 1, 2, 3. You can see it's all repeating units. And this one there, of course. And the sugar is a pentose sugar. It's 5-sided and it's actually called deoxyribose. And the next thing we're asked to do is to continue the second strand. So we're going to join the sugar, the phosphate, we're going to do the backbone and then add those bases in the middle as we work along. Then we're asked to construct a box around the code that would code for an amino acid. Now it's 3 nucleotides that would code for an amino acid. So I could take a box over those 3, or I could take a box over those 3, or I could take a box over those 3. So 3 nucleotides, 1, 2, 3, 3 of them code for 1 amino acid. And here we can see some work from one of my students. We can see where they have circled the nucleotide. Here's one chain. Here's the other chain running in an opposite direction. That one goes up and that one goes down. They've got the shapes aligned correctly so that they match. C to G, G to C, A to T, T to A. And here's the box where they've indicated the code for 1 amino acid. It's taken 3 bases, which of course are part of 3 nucleotides. Okay, so here's another worksheet here that I've used with my class. Looks a little bit more complicated, but it's really not. Here's my nucleus, and inside the nucleus I've got my double strand of DNA. It's all nice and loose, so we can read the code off. It's got a gap between it. Here's my nuclear membrane. It's got these nuclear pores, which are holes in that nuclear membrane. Here is the ribosome, the site of protein synthesis. And what's going to happen is a template is going to form on the DNA. It's going to travel out of the nucleus and it's going to rest on the ribosome. Now, when you move to A-level, you'll know that template is called messenger RNA. Inside your cytoplasm, we've got free-floating amino acids. They're represented by these circles here. The square molecules represent a carrier molecule known as transfer RNA. Here's one here, and here, here, and here you can see it's actually attached to the template. So the transfer RNA is going to move through the cytoplasm, pick up an amino acid, and carry it back to the template or the messenger RNA. This continues, and then as the amino acids sit next to each other, an enzyme forms a peptide bond between them. As more carrier molecules bring more amino acids, the chain gets longer and longer. And once each amino acid is attached to the next, the transfer RNA falls off and the polypeptide chain folds on itself and forms the protein. And the way it folds is dictated by the sequence of amino acids, which dictates at what angle and how many hydrogen bonds are sticking out and where, and therefore how it will fold, forming those hydrogen bonds between amino acids. So the first thing to do on the worksheet is to colour in the template blue. The template is shown in 3 places here: in the nucleus, passing out through a nuclear pore, and sitting on the ribosome. We are asked to colour the nucleus green, that's the nucleus there, and we're asked to colour the ribosome yellow. We're asked to colour the amino acids. We can see lots of amino acids. Some of them are free-floating, some of them are attached to transfer RNA, and some of them are already forming part of a polypeptide chain. We're also going to colour the transfer RNA molecules. If you're doing GCSE, you can just call these carrier molecules. We're going to label the DNA, we're going to label the nuclear membrane, and we're going to label the nuclear pores. It also asks us to indicate where we would find a peptide bond, and that, of course, is going to be between the amino acids here. The worksheet asks us to name the bases, A, C, T, and G, or you could say the full names: adenine, cytosine, thymine, and guanine. And it's 3 of those bases that code for that amino acid that is brought by that transfer RNA. 3 for that amino acid, and so on. So looking at that in colour, we can see we've got our template there, our messenger RNA, which sits on the ribosome. We've got our amino acids all over the place, including the ones in the polypeptide chain there. We've got the peptide bond. Here's another view of some other students' work. There we can see the carrier molecules, or we can call them the transfer RNA, in orange. Okay, another diagrammatic representation. It asks us to colour in the ribosome yellow. It asks us to colour in the amino acids red. That's our ribosome there. There are transfer RNA, or at GCSE we call them carrier molecules, and this is the protein chain, the chain of amino acids that are joined together by peptide bonds, and this is our messenger RNA here. This is our template that's been carried out of the nucleus, and that's what it looks like in colour. The ribosome's yellow, the messenger RNA green, the tRNA blue, and the amino acids red. Okay, so just to recap, we've got a pentose sugar, 1, 2, 3, 4, 5, and it's called deoxyribose. That's its alternative name. Attached to that, we've got a phosphate group, and also we've got a base, and there are 4 different examples of bases. The whole thing together is a nucleotide. So there's one example of a base: cytosine, thymine, guanine, and adenine. Guanine and cytosine will always pair together, and adenine and thymine will always pair together. They pair using hydrogen bonds, and between cytosine and guanine there are 3, and between adenine and thymine there are 2. None of that matters at GCSE. There's our polymer, and that's how they join. As you can see, we've got the bases in the middle there. They fit, you can see the shape. And you can also see that this chain goes up and this chain goes down. They're in opposing directions. And they then twist and form what's called a double helix. And you can see those bonds in the middle. They are the bases that are bonding. with hydrogen bonds. And these hydrogen bonds can be broken so that the DNA double strand splits, and at that point we can make a template against one of the strands. Here it's opened up and a template would form just there. Okay, so let's look at some past paper questions. A common question that's going to come up is when they give you a diagrammatic representation of the DNA.